Hello, everybody, to another edition of the Omics cast. And in today's episode, we're going to talk about how to set up a portable lab. My name is Alberto Vilella. For those of you who don't follow me on Twitter, my Twitter handle is twitter.com/Alberto Vilella, all one word. You can also follow me on LinkedIn if you want to ask me any questions or contact me. And this slide deck is available upon request. So in this episode, I'm going to analyze a paper, a recent preprint in bioRxiv, on how to set up a portable lab, a lab that you can take to the field. And the paper is entitled Rapid and Accurate Mean Ion-Based Workflow for Tracking Species Biodiversity in the Field. And the authors and affiliations are listed here. And the paper describes what is needed, the setup that they've put together for an end-to-end lab setup to do everything from sampling to sample extraction, DNA preparation, sequencing, and bioinformatic analysis as well. That's included in this portable lab setup. And they do it via an ONT mean ion, Oxford Nanopore Technologies mean ion sequencer in this case. And this portable lab features, as they claim, inexpensive instruments that can be carried as hand luggage and uses standard molecular biology protocols and reagents that tolerate the adverse environmental conditions. And they describe the different important aspects of how to do this and still get relatively good results out of it. And then they've put together a bioinformatics workflow that they call ON-Track, and we'll see what is that made of. And they say that they can identify, or that they require as few as 500 reads per sample to be able to identify what's in there in the field. And so they say that this is basically designed so that it's suitable for tracking species under field conditions. In the bioinformatics analysis, something that the paper mentions is the differences, the pros and cons of two different ways of analyzing the data that comes out from the mean ion sequencer. The reference-based approach, reference-based pipelines, they say are useful when matching a target sequence to similar existing ones, but because here they're trying to identify the biodiversity in the field, if you use a reference-based pipeline, it'll struggle to reconstruct an accurate barcode if the organism of interest has not been sequenced before, right? And so, for example, in the era of next-generation sequencing, if you want to align reads produced by a next-generation sequencing platform to the human genome, you just have to download a copy of GRCH38, for example, and that copy is a reference of the human genome that's complete enough that you'll get most of, if not close to all the information that you need. But if you're not sequencing the human genome, you're sequencing something else, then unless your reference based approach contains everything that you expect seeing in your reads, then you're going to miss out on what the reference doesn't include. And that's why they say the pros and cons of reference-based are versus the de novo assembly pipelines, and that they rely only on newly generated reads. These can suffer from more sequencing errors, especially if they are distributed in a non-random manner. And then they need to perform an ad hoc correction to be able to generate barcodes using this method. And so what they propose later on is kind of a hybrid approach that tries to get the best from both worlds. So let's focus on this Portable Genomics Lab that they've described in this paper, and they've shown the results from it. What is it made of? I've highlighted here 3 micropipettes, P1220, So they can go down to 20 microliters and up to 1,000. A mini microcentrifuge, a thermal cycler. This is probably one of the relatively expensive components. Electrophoresis system, a fluorometer, which in this case is a Qubit 2.0. It's probably also one of the expensive parts here. The nanopore sequencer, main iron, and a laptop. And they describe here the specs of the laptop. And they say they can put all these into a Peli case of 55 by 45 by 20, and I'll show a picture of this. And then they say that one special consideration here is temperatures. The required storage for certain reagents that can go from 4 degrees to minus 20 degrees Celsius, and they solve this by putting them in ice packs. The rest, they say, can go lyophilized and then resuspended with the relevant buffer and kept at room temperature. So the end-to-end process involves essentially using the PCR machine, and they describe here the primers they've used for this, and do a library prep, which involves ampere bits, for those of you who are familiar on how to purify DNA, and then quantified by the Qubit fluorimeter and then the Qubit DS DNA assay. And so a lot of this would not sound like rocket science to people that are used to the lab, but what's important is that they describe this system that can take place in the field. It doesn't have to be in a fancy lab to be able to do the end-to-end process. And then in the bioinformatics, after the sequencer has produced the data and the data is in this case, the laptop, they describe this on-track pipeline, where it's got some components that will sound familiar to most people used to bioinformatics tools like this. vSearch for rate clustering, then do some sampling, and then MAFT for multiple alignment, and they describe the parameters in which they do it, the local pair alignment method. A tool from MBOS in this case, which is used for producing a consensus sequence, and then they get this draft consensus sequence, and then they can realign read by read, in this case using, for example, Minimap, to this consensus sequence generated from the reads themselves. And then they use nanopolish at the end to the nanopolish consensus sequence. So as you see, they start with a draft consensus sequence and they produce a final polish consensus sequence. In this, if you want, hybrid approach, in which it's not reference-based and it's not de novo assembly, it's based on the clustering and multiple alignment process, and that allows them to iterate over and generate a consensus sequence. And so min-ion reads, we search for the read clustering, SIG-TK here for read manipulation or subsampling, generate a consensus sequence from the multiple alignment, and then the reads can be aligned to it and nanopolish to produce a final consensus, polish consensus. And so this is a little bit of, there are many components to this bioinformatics pipeline, but it's a very clever use of different tools that exist out there. Use the right tool for the right job and put it together into this workflow. And here's a picture of the setup, quite interesting. It all fits into this case. The laptop probably the largest, and they would say you can get good laptops today for, I put here an estimate, and it's this, is all very debatable, what type of laptop you need and how much money to spend, but $1,000 to $2,000. And then we got the Qubit to quantify, also $1,000 to $2,000, depending if you get it new or you get it from a lab that doesn't need it anymore. The PCR, quite small PCR cycler, thermal cycle for 1 or 2K to do the PCRs. Crucially, the sequencer, mean ion, $1,000 to $2,000, depending on how many devices and flow cells you want. And then the rest you can probably get quite cheap, either from directly buying them new or via eBay if you're not afraid of having to calibrate your own pipettes, etc. So quite a clever setup and quite a minimal system if you want to be able to go end to end. What's not shown here are the reagents, I mean, apart from the tips, but this would go in the inside the boxes and at different temperatures as required. I wanted to make a note at the end about how this combines into something that I'm starting to see more and more out there, which is I termed it DIY genomics, do-it-yourself genomics, which is now that you can produce, that you can devise these mini labs for not a lot of money. We're reaching a point where people can start thinking about doing their own do-it-yourself, do their own genomics experiments at home as private individuals, not associated to like a scientific project with budgets and all that, big budgets. In the case of many scientific projects, here is what can we do today? Where is the limit? What's the bottom? The minimum that you can do to do some genomics yourself, do some of your own genomics at home. And this table that you can find at the bit.ly/DIY genomics, DIY omics page that I maintain, I put here together different elements that I think people are probably going to muse about when they think about doing their own genomics. So the sequencers at the bottom and the nanopore fungal, and I put prices for the kit or the starter kit or the minimum that you need to buy and the unit price, right? So for a fungal, the unit price is at $90, but you have to buy a kit containing several fungal flow cells, and that would be a bit over $5,000. For the Illumina iSeq 100, a pack of 4 reagents will be $2,500 approximately, but each of those will cost you quite a lot more per unit than the fungal, not per unit of number of samples, but the unit of experiment, meaning in this case, combination of flow cell and reagents, right? And then above that, the mean ion plus a 12 flow cell pack would be $10,000 or $11,000, and then each experiment would be below $1,000, $800 or $900. In terms of compute and things that you can do immediately before or after sequencing, I put together here a few numbers and elements. Nanopore themselves, they sell the minute, which packages up a lot of this post-sequencing compute, and I'll set you back to 400, which is relatively cheap if you think about it, and it's highly programmable. You can add your own software in there. It's essentially a Linux box. And some of these in the middle, like the Intel Movodius or the NVIDIA Jetson Nano, etc. Some of these are, or the Google TPU board. Some of these are to accelerate some of this post-sequencing compute that needs to happen. And for Nanopore, the minute itself is essentially CPU-GPU combination, so you can do a lot of base calling in an accelerated fashion with the GPU. And some of these, the TPUs or the Intel neural network USB stick, are along the same lines. They're not a computer. They're something that sits next to a computer, is connected to a computer. In this case, it could be a laptop, etc., and allow you to accelerate some of these computational aspects that can happen post-sequencing. And this doesn't need to be only base calling. It can be all sorts. It can be your machine learning for whatever you want to do, cluster it, etc. I put together here also some of these mini computers and things that look more like a mini motherboard that can have other connections, like the Raspberry Pi or the Adafruit kit, and Arduino starter kit, or this Kano computer kit touch. These are quite cheap, as you can see, all of them, but it'll give you flexibility in tailoring or doing more bespoke computational rigs if you really know what you're doing. Obviously, this requires a deeper knowledge on not only the software but also the hardware and the electronics behind. And then on top, I've done a couple of them on sample prep. The Voltrix itself from Nanopore, which my calculations make it at a per unit price of below $200, but with a kit that contains a lot of things, including sequencing reagents and mean ion flow cells. But if I make my calculations based only on the part that's strictly related to sample prep, then each unit of experiment could go down to 179. And the open drop is the kind of do-it-yourself electrowetting system, and it's below $1,000. So if you go to the website, you can click on these links and find more information about it. And so I wanted to make this connection between this paper that relates what's the minimal portable lab that you need to do something useful in genomics, with what are the possibilities out there to have your own genomics set up, as a private individual. And so here is the same as before, just plotting the kit and unit prices for the different elements I described before. And then the questions or the challenges here are we at the point where someone can devise a DIY genomics rig for, let's say, $5,000, including some sequencing equipment, some compute equipment, some sample prep equipment, DNA quantification equipment. And can someone come up with a DIY genomics rig where the experiment unit cost each time you want to do a sample is around $100 or $500? And so I don't have an answer to these questions. I can put some of the pieces of the puzzle together, but I'm starting to see this puzzle being more and more complete. And this paper about the portable lab is an example of people thinking along those lines. What's the minimum that you need, in this case, to make it portable, but also small and minimal to do some genomics, right? And that's why I wanted to make this connection. I hope you enjoyed the analysis of this paper and the DIY genomics analysis I made. And as a reminder, you can follow me on Twitter on my account, twitter.com/Alberto Vilella, one word. And you can follow me on LinkedIn if you have any questions or anything that you want to discuss privately. Please do so. This slide deck is available upon request. And thank you very, very much for listening to another episode of the Omics cast, and I'll see you in the next episode.